11 a qué? ¿A 12?
A 12.
Ah, bueno.
Bueno,
Sacar rápido.
como a 11:50 para que haya preguntas.
Sí.
No tanto. Buenos días a todos. Bienvenidos a los estudiantes, facultad, tanto del departamento como de otros departamentos de ciencias médicas y personal del CDC. En el día de hoy tenemos a la doctora Rebeca Rico, que nos va a hablar sobre el dengue, específicamente la epidemiología y la virulencia del dengue. Anteriormente la presenté, pero como tenemos personas nuevas en el seminario, pues la doctora Rebeca Rico se graduó del bachillerato para los años. Ay, no digo los años, ¿verdad? No. Hizo en la Universidad de Nebraska. No sabía que tú estabas en Nebraska. Tengo colaboración con Nebraska. Y entonces hizo maestría en salud pública y luego un doctorado en virología en el Cornell y luego un postdoctorado en el CDC en Atlanta. Ahí fue donde conocí yo a Rebeca, que estaba yo haciendo el doctorado en Emory, haciendo research en el CDC, y hicimos muy buena amistad. Rebeca fue profesora asistente en Yale, en el departamento de epidemiología, luego profesora asociada en Yale también, y se encuentra desde el 96 trabajando como científica en el Departamento de Virología e Inmunología en el Southwest Foundation for Biomedical Research en San Antonio, Texas. Rebeca ha obtenido muchos honores desde el 77 hasta el presente. Entre ellos se encuentra el Charles Shepherd Science Award, que es uno de los awards más distinguidos que se le da a los trabajos de investigación en el CDC. También ha sido chairman de comités de Arthropod-Borne Virus. También ha trabajado como consejera, ¿verdad?, en World Health Consultant en el World Health Organization, WHO. Y también ha salido, además de tener muchas publicaciones en el campo de poliovirus, genotipo, en secuenciación, molecular evolution de dengue. Perdónenme mi Spanglish. Evolución. Ha trabajado mucho con virus también hemorrágicos. Fue una de las descubridoras de los virus que dan hemorragias en Venezuela, ¿verdad? Juanarito. Y nos habló mucho sobre esa epidemiología y cómo nosotros no debemos estar en la conferencia anterior muy asustados con la prensa, porque muchas de estas enfermedades pues, se mantienen localizadas en esas regiones y usualmente resurgen, son emergentes porque nosotros somos los que cambiamos el ambiente, no tanto como que ellos son virus nuevos que cambian y mutan y se diseminan rápidamente. Esto lo vamos a ver hoy con el seminario de ella sobre la epidemiología del dengue. Con ustedes la doctora Rico.
Muchas gracias. Unfortunately, I'm going to have to switch to English. Yeah, it's easier. It's more efficient because I don't think I want to do Spanglish while I'm being recorded or Pocho. Is this? No. Okay. I'll try to stay away from the. Yeah, there we go. Thank you. I'm going to talk about dengue right now. We had a workshop earlier with the students, and I was trying to explain what emerging diseases are and how viruses really aren't as nasty as they're portrayed in the press. Especially right now, we're having an outbreak of Marburg in Angola, and I don't know, Ebola seems to be making it into a lot of movies, but we should be more worried about things like dengue that have existed for many, many years that are affecting people, millions of people each year throughout the world. And I guess it's only because it hasn't entered the United States recently is that we have not as much interest, especially when it comes to NIH funding. Right now, we're lucky though in that dengue got on the list of priority A agents that, so that there is better funding from NIH, but as a bioterrorism weapon. And really, I don't have the slightest idea of how dengue can be used as a bioterrorism weapon. Actually, I read a proposal that was very creative. It was very creative. They said, well, what if if people inoculate a bunch of mosquitoes and liberate them? And I'm like, how are you going to do that? Are you going to put them on a plane and then dump them out? And I don't think the mosquitoes can adjust to the differences in pressure and all these things. So anyway, people are trying to make up new ways of making it a bioterrorism agent, and thank God there's really no way. But we already have enough of a problem, okay, with dengue, and that's the point I'm going to try to make. So I'll go right to it. Dengue belongs to the family of flaviviruses, and has a positive strand RNA as its information, genetic information. There are 4 different serotypes. That means that you can distinguish 4 different viruses based on antibodies. I know there are people in the room that don't do this all the time, so excuse me for repeating some of the basic things here, but it's to show you how there's a diversity of viruses, and even mice can distinguish those viruses by their antibodies. Okay, mice are obviously not a natural host of dengue, but dengue is right now maintained in a human to mosquito cycle. There are supposedly still some enzootic cycles in Southeast Asia and Africa, but there's been no recent evidence of primate mosquito cycles. Those viruses, they haven't been isolated since the 70s, so we're not sure if they're still there. And right now, it's become more a problem of urbanization, where you have the mosquitoes living close to people and biting only those people in that house. So it causes classical dengue fever, which is a flu-like syndrome, but it also causes a hemorrhagic disease. And that's the one that has proven fatal to many infants in Southeast Asia, where they have a long, long history of having hemorrhagic dengue. But now in the Americas, we know that it can kill adults also, okay? Internal hemorrhaging. And this is to be distinguished from other viral hemorrhagic fevers that actually kill people a little bit quicker. And, oh, thank you. Was that? Yeah. It kills, other viral hemorrhagic fevers kill people quicker, and the bleeding is much more massive. For example, this Marburg and Ebola and some of the arenaviruses that I've worked on in the past, there's massive bleeding. A person can die in a matter of 24 hours. Dengue hemorrhagic fever is a little bit slower, and you have to go through the classic dengue fever before you go into the second step, the hemorrhagic part. And as I mentioned before, it's a really big problem. 50 to 100 million infections per year. And a review that we just read this morning says that there are approximately 500,000 cases of hemorrhagic fever per year. And of those, approximately 20,000 die. Okay, so it's very underreported, at least in the media, or people in developed countries are very, very much not aware of dengue. So, and it's responsible for explosive outbreaks that, as you will see, are usually due to changes in people's habits. We're the ones that are creating the situation. We're the ones that are getting the mosquitoes to breed, and we're putting ourselves in that cycle, that natural cycle of disease. Aedes aegypti is the main vector. It's a domesticated mosquito. It has adapted to living with people. It prefers to bite people. It's active during the daytime, and it's in any place you can find water around. Flower pots, stored water, used tires, tin cans, anything that will collect water. In San Antonio, we have a real problem with bird baths. People that don't clean them out constantly, there'll be the larvae in there. So this is a problem throughout the eastern coast of the United States, and obviously in the Caribbean and all the rest of Latin America. So right now, people in the United States are not aware that we have very high numbers of the vector. We're just waiting for the virus to get into those mosquitoes in the United States. Okay. It feeds frequently on people, and the eggs and the larvae are in all those containers around people's homes. Then the adults come in, and they can hang out in your closet, underneath the bed, and they come and can bite you at night or during the daytime. In fact, people that are out doing research trying to collect adult mosquitoes have actually gotten infected when they're doing the research. Okay, and we know that because a really good student of mine from Yale is out doing this in Iquitos in Peru, and they're actually tagging the mosquitoes, and they're looking at what the mosquitoes, who they fed on, and they can actually trace it back to some of their workers versus the people that live in the house. So this is very delicate work. Let's see, there we go. So this is the world distribution of the disease, but as you can, the yellow, there's more yellow here, because that's the distribution of the mosquito as we know right now. I think that if we had more entomologists like Roberto out there, there'd be more yellow all over the place. If you have the people doing the research, you find the mosquito. Okay. And we do have reports of disease in many of these areas, but there's still some that are just waiting for things to happen, like I said, in all this area of the United States. There's tremendous potential. And if it does happen, remember we have a large population of non-immunes, so things will spread very quickly. The only thing that's different in the United States because we have experience in Texas, where I'm living right now, we have the experience with the border with Mexico. The only difference between the two populations across that border is that in Mexico we tend to hang outside and talk with people, and in Texas they're watching TV inside the house. So there's a screen, there's a barrier, and it's a matter of habit. Also in Mexico, some people can't afford to have the screens, so they may be inside and not, but still get bitten. So we can have an outbreak in Reynosa, in Matamoros, and hardly have any cases across in McAllen and Brownsville and Laredo, Texas. It has happened though, and there have even been deaths. But so far, no hemorrhagic dengue. There haven't been enough infections to get to the hemorrhagic stage. And we've been looking at viruses from across the border in Mexico, and they're of the non-virulent type that I'm going to describe to you. So the potential exists for dengue to occur in many, many areas, and as you can see, it's just a whole wide band that is pretty much the determining factor of how far the mosquito can go. Okay.
What is the island of the coast?
I'm sorry.
Chile?
Yeah.
And it had a dengue activity there.
I guess if that was red, Dwayne Googler did this one, so he knows.
But there's,
I don't know personally. I don't know personally. I'm sorry.
human population there. So,
I'm sorry. I have no
the
information.
orange.
Wasn't all orange, and I think that's because it's probably too small. They want you to see that there's plenty of potential, but we know that there is, and it's not constant, but there is. Okay. So I'm going into the clinical classification right now, just to highlight why it's important for us to get the clinical information in addition to what I'm going to show you that we do now, more the molecular virology, because you need to make sure that this is dengue that you're looking at, that's what the person's sick of, so that when you go out and you start doing sequence comparisons, that you know that it came from a case of dengue or hemorrhagic dengue. And my issue with this is that you need all these lab tests to confirm that a person had dengue. And what it means is that you're going to have dengue classified or diagnosed where you have a lab. And if there isn't a lab, dengue isn't going to be there. Okay? So it's a false sense of safety that we have here. Until you have an outbreak of large enough, usually dengue hemorrhagic fever, do you get enough laboratory backup or attention from outside, or get samples to a reference lab elsewhere to get all of these tests done. But my main point is that for hemorrhagic fever, you have to meet these very specific criteria. And I mention this because in the 1970s, supposedly we had dengue hemorrhagic fever cases, sporadic, but dengue hemorrhagic fever cases in the Americas. But this information was not along with it. So it doesn't really meet those criteria, especially the thrombocytopenia or some of the other very specific, the rise in hematocrit. And it's just things that you need to meet, that they have a lot of experience with in Southeast Asia. But when dengue hemorrhagic fever has occurred in new places, there's very little experience as to how to classify it as hemorrhagic. Okay? And that's very important, as you will see. Then there's the shock syndrome also. But this seems to happen more in children and infants in Asia. And again, things have changed a bit. In the Americas, we have some of the more severe disease in adults, and it's not clear why that is, even though they've been exposed to other serotypes of dengue. So again, very complex type of disease that can manifest itself in different ways. Now, the reason why you go into the hemorrhagic stage is very complicated. We know that people, you can have, because there are 4 different viruses, remember I said there are 4 different serotypes of virus, you can be infected 4 different times. Okay? You won't get reinfected by the same serotype, but you can be infected 4 times with dengue. And as you go, the possibility of you having a more severe disease, i.e. hemorrhagic fever, increases. And this is supposedly due to antibody-dependent enhancement of infection. Unfortunately, that's only been shown in vitro, in cells in a petri dish. And that is the main problem with dengue. We don't understand the pathogenesis because we don't have an animal model of disease. Right now, there still is no animal model of disease. Even higher primates, even chimps, gorillas, do not develop the disease. They have the virus in their blood, but they don't get the rash and the fever and even the hemorrhagic manifestations that we humans get. There's also increased T-cell activation and cytokine production. You can measure the cytokines in the patient's blood. Tremendous elevations. So it's a basic inflammatory process. But we don't know how that contributes to the pathogenesis of disease. We know that there's major plasma leakage, but the endothelial cells that line our circulatory system are not really infected per se. They do not produce virus, but they do respond to this inflammatory process, and their junctions separate, and you get plasma leakage. So that's what starts this whole issue of the internal bleeding, the hemorrhaging. So because we don't have animal models of disease, we've been trying to work many different aspects of dengue to understand its pathogenesis. And the thing that worked for us in my laboratory was to put the epidemiology together with the evolution of the virus. Okay, so that's the only thing that made us a little bit different from everyone else, is that we took sort of like the two extremes, what's happening in the population as a mass and clinically, and then the molecular biology, looking at the genetic variability of the virus. So linking those two things showed us that there are actual differences. There are specific viruses that produce hemorrhagic dengue and others that do not. But it took over 20 years of research to show this, because we're sort of beating around the bush instead of directly addressing the thing. We cannot inoculate viruses. We cannot inoculate animals. I'm glad I corrected myself. Thanks for the replay. The first differences in viral virulence were shown in the South Pacific because there was less virulence, not because there was more, there was less. And Dwayne Gubler, who used to be here at CDC, was the one that did those studies. So let me just go to a very complicated evolutionary tree. And this is basically the most up-to-date evolutionary tree. I don't expect you to see all of this, but it took us 20 years to reach this point. Okay, and it used to be that I would show these how can I put it, very primitive trees before, and now this one has, I don't know, something like 60 different viruses in it, and I selected those to just be able to give you better examples of what's happening. And what this is a comparison of the glycoprotein gene sequences of about 60 different dengue serotype 2 viruses. And we have a sample of each one of the other serotypes down here at the bottom. So we have a dengue 1, we have a dengue 3, and we have a dengue 4. Just to show how distant they are from all the other dengue 2 viruses. And I have shaded the regions or the groupings as to genotype. Right now, we describe dengue serotype 2 viruses as belonging to one of 4 genotypes. So the bottom one here, viruses from the western area of Africa. And these viruses are actually maintained in those enzootic cycles between different primates, monkeys, and different types of mosquitoes, not just Aedes aegypti, but let's say the African version, some African Aedes-like, aegypti-like mosquitoes. And those viruses are not infecting humans. There is one isolate here, one virus that was sequenced that we actually got from a 14-year-old girl, but she actually went to a clinic because she had malaria. And they took a blood sample and then they isolated the virus later. But she wasn't really suffering from dengue. So we think that these viruses may actually be attenuated, that they're not producing outbreaks. We know that they're not producing outbreaks, but the people in the same countries where these enzootic cycles exist, for example, we have Senegal and Ivory Coast here, they actually imported their epidemic viruses from other continents. So we have samples from Ivory Coast, I think it's up in here in the Southeast Asia group, that were actually imported and produced the epidemics. So yes, the ones that are being transmitted in the monkeys and Aedes aegypti-like mosquitoes in Africa are not doing anything to the population, to the human population. And it's actually very hard to detect these, or to get these samples. And the people from the Pasteur Institute are the ones that are out there actually working in the jungles, isolating the viruses from mosquitoes. Then we have a second group, which is the West Africa viruses are the most distant. They're very different from all the rest. And then we have a group of 3 different genotypes that are a little bit closer related. But we have a group here that I just called them from a genotype of the Americas. And it actually spread to the Pacific also, to the South Pacific. And these viruses have been in the Americas apparently since the 1970s. We have one strain here from Puerto Rico, and I think it's 1969. And there was one isolated in Trinidad back in 1953. So those viruses have been in the Americas for a very long time. But now it turns out that this genotype from Southeast Asia has actually come to the Americas, and it has displaced these from the Americas. And this genotype from Southeast Asia is actually the only one of the 4 that produces hemorrhagic fever. Okay? It's taken us this long just to be able to say that. Only the Southeast Asia genotype produces dengue hemorrhagic fever in humans. And the ones that we had, that we call it Native American genotype, has never been isolated from anyone that meets the criteria of having dengue hemorrhagic fever. And we now know that in northern Mexico, when we get samples from across the border from us in Texas, the American genotype still exists there. It still exists in areas of Peru, where people have looked, in Peru, northern Mexico, and maybe some other areas. But this, if people have outbreaks and they take samples from patients, and if there's any dengue hemorrhagic fever associated with it, it has always been Southeast Asian genotype. Okay, and there is one more genotype that doesn't seem to be really that active. It occurred in Asia and Indian subcontinent, but actually they have imported the Southeast Asian genotype too. Now India has dengue hemorrhagic fever. Their sort of native genotype may be being displaced by the Southeast Asian one also. We don't know yet. But they're detecting more and more of the Southeast Asian. Okay. So the problem we have right now is how many of these other genotypes is the Southeast Asian going to displace? Is it going to go as far as the West Africa? Is it going to invade the African continent and now produce dengue hemorrhagic fever, where there has never been any? And that's going to be up to those mosquitoes, whether they're going to be capable transmitters. And the entomologists are going to have to help us with those tests to see what it is about the mosquitoes that make them more efficient vectors. What makes them more efficient is to transmit this virus that produces more disease. We've done some of those studies in our lab. But this is just to go back and go through some major points, and then I'll go to the mosquito work. We have demonstrated how the virus can jump across to other continents and infect and disperse through the Aedes aegypti populations in other continents. That dengue did not originate in Africa. The most virulent actually came from Southeast Asia. Okay. There's a separate genotype that exists in Africa, but so far there's no real evidence that it came out of Africa. The one that is producing disease in Africa actually came from Southeast Asia also. Okay. There's another controversial issue right now. There have been several publications saying that dengue undergoes recombination. That is, that the viruses get genetic information from other dengue viruses. But as I explained earlier, for several reasons, that hasn't really been documented well. That's all based on statistical analysis of sequences. No one has actually isolated the virus and shown that it's a recombinant. But there are over 20 publications out there saying, watch out, dengue is becoming a real problem because it's recombining. And unfortunately, everybody's talking about that again. The media loves that type of story. But they have not met the classical virology criteria to demonstrate that there's recombination. And so far, there has not been any increase in virulence in these so-called recombinants. They haven't done anything out of the ordinary. Okay. Again, this is just a lot of information, but what it is to show you that we have another way of trying to get at this issue of, okay, so what makes the Southeast Asian genotype more virulent than all the others, or what makes it different? And what we did was we got patient sera from several countries in Latin America and from Southeast Asia, and we amplified by RT-PCR, just a technique to make enzymatic copies of the RNA of the virus, directly from the serum of the patient. And we got the sequences of the whole genome of the virus in that person's blood sample, of the whole virus. And we compared those virus sequences from people that had an American genotype infection to a Southeast Asian genotype infection. And we saw that there were specific areas of the genome that were always different between those patient samples. And I've shown here the amino acid differences, but just to summarize, there are some differences that are important in terms of charges. That is, the amino acids that are consistently different have a different charge, and that may make a difference in the structure of the proteins that the person's immune system sees, or it may make a difference in the receptors, so that the Southeast Asian virus can attach more efficiently to the macrophages and dendritic cells that it normally infects. And the American virus may have just less efficient receptor and attachment processes. And the other thing is that there are differences in the regions of the genome that do not code for amino acids. These are the two extremes of the RNA and how they fold. And that, to us right now, is probably the most significant difference, because the way that the RNA folds is actually determining how fast the virus replicates. And that right now is our focus. This is just to show you differences in the 5' ends, this very short area of the genome on the left extreme, that the way it folds and how it may make a difference in the way that the polymerases come in and attach to the RNA to replicate it, and also how it interacts with a 3' end. And I had mentioned earlier, we're now doing mathematical modeling of how the RNA of these different viruses actually folds back on itself. And these are areas that don't code for any amino acids, but the American viruses, let's see, A and B. Yes. The American, is A. The American viruses are not as convoluted as the Southeast Asian viruses. The Southeast Asian viruses seem to fold back on themselves a little bit more. And apparently that makes the replication complex. These parts actually go back and fold back with the last slide I showed you. So they curve around, the virus curves around the 5', sticks together with a 3', and there's a polymerase complex that makes copies, and that determines the efficiency. So maybe the self-folding of the 3' end may make that replication, or the beginning of replication, of copying the RNA much more efficient. But all this is theory. And again, even these models are theory. This is just, we haven't perfected the way of knowing the folding patterns of RNA yet. So what we're doing now is making infectious clones. That is, molecular infectious clones. We're taking those amino acids that we saw were different, 5' and 3' ends, and switching them out. So we'll take a Southeast Asian virus and stick in the American genotype parts, and then see how they replicate. And we can do the reverse also. Have an American genotype infectious clone and stick in the Southeast Asian and see if that increases the replication rates. And they do. Of course, that took years to do. And I'm just switching through the slides. But this is just a graphic to show how if you take a Southeast Asian virus and you stick in the American genotype structures, the 5' end, the 3' end, and that one amino acid in the E-glycoprotein, all just those three structures, if you stick them into the Southeast Asian genotype virus, it starts acting like the American virus. You decrease its replication rate. And what we have here, the last two bars here, represent our infectious clone with the 5', the E amino acid, and the 3' inserted with an American genotype virus acting just like its own wild-type virus in terms of replication in dendritic cells in vitro. Okay, that's the best thing we have right now, dendritic cells, human dendritic cells that we actually extract from Buffy coats. We get anonymous donors donate a pint of blood, we get the Buffy coat, we grow the monocytes out, we purify them, we stimulate them to mature into dendritic cells, and then we infect them with virus. And this is the closest thing we have right now to showing how the virus affects humans. And what we did was we took a collection of 24 low passage viruses. And this is the difference again in my lab. We're working with very low passage viruses. We're not working with the prototypes that people work with because they grow real easy. The prototypes, there's one called New Guinea C that's been passaged over 20 times that everybody works with. It's easy, it grows like a weed. But it turns out that it doesn't really reflect what the low passage viruses are doing.
And are the titers of your primary isolate as being high as
Yeah. Well, enough for us to be able to do the infections. And we don't plaque or anything like that. We just do TACMAN. We do quantitative RT-PCR to keep track of the numbers of viruses, because it also turns out that low passage viruses, when I say low passage, I mean less than 5 passages in vitro, less than 5 passages in cells. They don't plaque as well. New Guinea C and this other 16681 that's also used as a reference, boy, those, we call them gangbusters. They go out and they just take off, and they produce tremendous numbers of plaques, and they're consistent. But these do not. So I had a really good postdoc, Ray Colonna, go in and take 24 different low passage isolates, patient isolates, and he infected these monocyte-derived dendritic cells from a whole bunch of different donors. And what we have here is a comparison of different donors. And as you can see, people's dendritic cells will replicate the same viruses at different levels. So that's telling us, okay, there are human differences here. And these are all donors from San Antonio, Texas. We haven't had dengue in San Antonio, Texas, okay? So they haven't seen this virus before. They don't have any immunity. And actually, these cells have been, how can I say, purified away from any antibodies that these people might have, okay? So this is natural reaction to infection, innate immunity of the person's dendritic cells. And we have, look at the two columns here versus this donor D down here who's barely replicating the virus. So something's going on.
Is it, is it the level of receptor or intracellular?
No, we don't know that yet. That's going to be hard to do. We still don't know what the receptor for dengue is. There's some papers out there, but several receptors. Well, just like HIV, there's several receptors. Yeah, co-receptors, right?
Is that the genetic cells from the people who were infected, or is that the genetic cells from both codes?
These are just cells from people that have never been infected with dengue, anonymous donors. This is an innate immune thing. That some people's dendritic cells will replicate dengue to other degrees, to different degrees. And that one's going to be really hard to understand. The other thing is actually the receptors aren't so much the issue based on some other studies. When Ray does FACS analysis of these dendritic cells, actually the level of infection of cells by this donor D is almost the same as donor A by FACS. Most of donors, let's say 30% of donor, these cells are infected, well, so are 30% of donor A's. The problem is that donor A is putting out a lot more virus than donor D per cell. Yeah. Okay. So there may be a receptor issue, there may be a co-receptor issue, but this is just some sort of intracellular. Yeah. Well, at least we're seeing it expressing viral proteins by FACS. Okay. But the way that they're pumping out virus is a different deal entirely. So, so the other thing we did was we compared. Well, anyway, in these, the bottom line is that we show that the Southeast Asian viruses replicate or produce much more virus, much more efficiently than the American genotype viruses. Okay, so besides the differences in the way that people get infected, there are significant differeces in the way that the American genotype, the Native American virus that we had here, replicates in dendritic cells in vitro versus the Southeast Asian viruses. And then if we break it down and we go into when is this happening, it's happening at the beginning of the replication cycle at around 12 to 24 hours. Basically, by 12 hours after infection of these dendritic cells, there is a tremendous difference in the way that the Southeast Asian virus produces particles than the American genotype virus. So it's an immediate thing. And we looked at negative strand RNA synthesis versus positive strand RNA synthesis to see if it was, at what level, that it has to do with replication. It has to do with that folding back and getting replication to started with a polymerase coming back and making copies of the virus genome. By 48 hours after infection of these dendritic cells, both the Southeast Asian and the American virus produce the same amount. But that makes a difference if you're talking about a person. The person that's infected with a Southeast Asian virus is already pumping out tons of virus in the first 12 hours. Okay, if we convert this into a whole body. And again, the other thing that we wanted to look at was, okay, some of the viruses that we studied in this collection of 24 had been isolated from dengue hemorrhagic fever patients. And people had always thought that it was an issue of the virus. And if it came from a hemorrhagic fever case, then that virus was more virulent. Turns out that is not the issue. The issue is whether it belongs to Southeast Asian genotype or American genotype. And that we showed by taking the donors, and And looking at the difference again of if they started out having DF or if they had DHF. And there's no significant difference. The DHF samples have a little bit more, but it's not statistically significant. Okay. So there's nothing innate about the virus that's producing something in the person that has dengue hemorrhagic fever. It's something innate in the person that's making them replicate virus, plus that they have to start out with Southeast Asian genotype virus. American virus is not going to replicate. Okay. So I have to do this quickly. Last slide here. So the other thing we want to look at is what's happening in the mosquitoes. Are the mosquitoes also making a difference in terms of what's happening? Why is the hemorrhagic fever, the Southeast Asian virus is much more efficient? Are they being transmitted more? Are they, what's making them displace the American genotype in Latin America? Turns out that the Southeast Asian genotype viruses, again, are much more efficient at being transmitted in mosquitoes. These are mosquitoes from Macallan, missing an N there. Field collected mosquitoes, well, actually generation 1, after they collected the eggs in a field, you grew up the mosquitoes and you infect those mosquitoes, and they're much more efficient at infecting, disseminating the virus. Okay. They get infected easier, and they replicate the virus easier, and it gets into their salivary glands faster. Okay, so they have a tremendous advantage in the mosquitoes also. And these are mosquitoes that were collected, well, the eggs were collected in Tehuantepec in Oaxaca in Mexico, just to show that two different populations of mosquitoes, Aedes aegypti, collected in South Texas and Oaxaca and southern Mexico, are capable of showing the consistency in the way that they transmit. It's still, Southeast Asian virus is much more efficient. And then we did another study. Well, Phil Armstrong did this work in a postdoc in my lab. This is very difficult stuff to do. He dually infected the mosquitoes. He took mosquitoes from McAllen, Texas, went out and collected some more, infected them with both viruses at the same concentration to see what happens in one mosquito. Okay? Does the Southeast Asian win over the American genotype? And yes, it does. The bottom line here is that he competed several different viruses. He took one Southeast Asian and then this strain of American virus. So he did 9 different co-infections and found that at the end, 21% of the mosquitoes had the Southeast Asian virus outgrow the American virus. And only 3% had, only 3% had the American genotype virus in them. And some even had both. So if recombination is going to happen, it might happen in the mosquito. But it still hasn't happened. And so that what this is telling us is that in nature, the Southeast Asian virus is so efficient at getting into the mosquito, replicating, and getting transmitted back to humans, that's why it can produce these epidemics, and that's why it's going to displace the American virus. Even if they both infect the same mosquito, Southeast Asian is always going to be ahead. Okay. And I'm running out of time here, but I just want to mention one thing that we don't have even tabulated. We're still working on it, and we're preparing a manuscript right now. We think we may have an animal model for dengue fever. What we've done is another weird thing, chimeras. We've used skid mice, severely immunosuppressed mice, and we have engrafted them with human stem cells that have been purified. And these are white blood cells. And what happens is that the mice actually develop full-fledged Langerhans, human Langerhans cells. And these mice even get a rash. Not only do we have a viremia, they have a rash, and it looks like they're making antibodies. And these are just transplanted human dendritic cells. Unfortunately, it's real hard to make these mice. We work with some people in Dallas. They're non-skid, they're gamma irradiated. You have to put human cord blood into these mice. Wait for 8 weeks. And then they have to test their blood to see what the transplantation rates are, and they're pretty good. It's sometimes, more recently, they've given us mice. Of course, these mice are more valuable than people right now. We work with 6 mice at a time, and we're lucky to get 6 mice. But they have up to 40% of their white blood cells are human. Okay? So it's amazing. We finally have something that we think. So the rash this is the first animal ever to have we have photographs. We use this erythema meter that dermatologists have. We do all these really funky things with these poor mice. But we're preparing that manuscript right now. We're still analyzing some stuff that Idali's going to help me with. So we hope this will be the future. We hope that we'll later be able to infect these mice with other strains of dengue and see if this antibody-enhanced infection business is true. Yeah, a lot of things that we can do. We've tried measuring cytokines, but with 25 microliters of mouse blood, you can't detect cytokines. They're so tiny also. Yeah, not enough.
So they're making tests for multiple cytokines.
Yeah, we tried it. We did that. We're working with a company. It's in their best interest. It's in their best interest. But anyway, that's the future. I hope that we might have a model that we can really start doing a lot of things with. And when these low passage viruses that we have fully sequenced, all these other things, and link it with the mosquitoes, then we can have the mosquitoes transmit to the mice and all kinds of neat things. So hopefully I'll be talking to you about that the next time. Thank you very much. Yes.
When you originally saw the American genotype and compare with the Southeast Asian genotype, did you ever look at viremia in these people? So does viremia really correlate with disease severity in the human population?
Okay. Yes, actually, that was back when we were working on a program project with Frank Ennis out of the University of Massachusetts. They were doing clinical studies. It's a very large program project that was being done in Thailand. And David Vaughan did all of those studies, and some of those were included in that chart, that full genome sequencing that we did. And yes, there is a higher viremia in dengue hemorrhagic fever patients. It does correlate. Was that your question?
So when comparing Southeast Asia genotype with American genotype?
Oh, no, we don't have, unfortunately, we don't have prospective studies where you have both happening at the same time to get the clinical information and sufficient people to have both, because, as I told you, it seems that one Southeast Asian comes in and it dominates. All the people that get sick have Southeast Asian. I don't think we're going to have any specific place where you have both happening at the same time and then look at viremia. Although you could do a prospective study in northern Mexico just on the American and try to extrapolate, American genotype, I mean, and try to extrapolate. But Dwayne Gubler back in the 70s, that South Pacific stuff that I mentioned before, showed that those viruses that were American genotype in the South Pacific in the 70s produce lower viremias. But we don't have that right now from the Americas. Yeah, that's unfortunate. We need some prospective studies. There are some going on right now.
So there are sequence differences between the two genotypes
in
the non-structural region.
Yes.
Have you looked into these regions at all?
Well, we did the infectious clone stuff to show that if we put those things back in there, yeah, it decreases.
No, but that's the flanking region.
You mean non-structurals like the 1 through 5? We have the next set of mutations that we have to put in are the NS5. NS5 has several, about 3 amino acid differences, I believe. We don't know how important they are, but by just putting in the 5' and the 3' and the E, we're already getting back down to wild type. So we have to do the opposite and see if we can get it back up. But NS5, I'd say, would be the other potential. It's a polymerase. But we have those on the back burner.
My last question, I'm sorry, I think. But you did these experiments in dendritic cells. Have you looked at the dendritic cell maturation? Is there any difference? I mean, presumably, if they are better infecting, the dendritic cell will be better antigen-presenting cells. So how does that?
Actually, dengue only infects immature dendritic cells. So the maturation is occurring during the infection process. And no, we haven't looked at that in detail. We haven't gone through and do fax analysis, that often. I mean, Ray did a really good job of going every 12 hours and things like that. But we need to do some more detailed work on mechanisms. Yes, but I guess right now I'm too excited about the mouse studies. Like, wait, everybody do this right now. Yeah, but we'll get back to that. Thank you.
When you did this infectious prompt, did you find that one region was predominantly more responsible for the
Oh,
infection? Was
I
it
see.
envelope?
No, it was. You can't say anything like that. It had to be all three. Each one decreased a little bit, decreased the replication rate a little bit, but it's only until you have all three together. The 5', 3', and the E, that you get down to wild type. It's synergistic. It has to work together. Yeah. Yes.
Yeah, my question is, do you think that this 5 and 3 UTR regions might be affecting the stability of the RNA?
The stability in terms of, you mean the replicative form, or?
Yeah, that the American is becoming debated quicker than
No, because during replication you have this massive polymerase, with cellular factors and stuff sitting on it, and remember, it's a double stranded because it's negative strand that it's making. Yeah, it's positive when it goes in. It makes messenger, makes stuff to make up the negative, then it starts just pumping out negative, negative, negative to make more positive and positive. So we don't know exactly at what stage all of this is happening, but we know that once it's internalized. And I'm working with Eva Harris of the University of California at Berkeley to try to break down the mechanisms to see what transcription, translation, things like that. It's very difficult work. She's working on one virus already, a Taiwanese virus, that she has things kind of broken down, that she thinks it's a translational control. But transcription is really hard to address, especially you get that virus in there and within minutes it's already doing transcription, and you basically have gotten your reagents together, and you're still trying to work that out. Yeah. Yes, sir.
I've got a question. But the main thing would be if you could not be bitten by a mosquito. Is there any, I hear a lot of OYS tales and you eat garlic or something like this, and the mosquito is unattractive to you. Is there any truth in anything?
I'll let Roberto answer that. Roberto is an entomologist. I've known him for a long
The
time.
population was being bitten, apparently.
Yeah.
Maybe there were some that weren't being bitten, probably.
Did you want to answer? Whether there are differences in attraction of human hosts to mosquitoes. And of course there are, but how can you modify your attractiveness to be bitten?
There are many more questions in that area than answers.
Yeah.
We do know that there are several odors that attract mosquitoes to the host. And went to last week to American Mosquito Control meeting, and there's a company that is developing a bait for Aedes aegypti, and the guy gave me so I could smell it, and it was awful. Just a cocktail of human odors coming from different parts of the body, and better for the mosquitoes.
So take a shower. It's a complicated story. Shower often and forget about the repellents. DEET, I know. If you're out in some tropical areas, it's on you for 10 minutes. That's it. Yes.
¿Cuál fue el serotipo que entró a Cuba en el 81
Ah,
y a
sí.
Venezuela en el 89? ¿Ya ha cambiado eso?
It's the same thing. It's Southeast Asian serotype 2 virus that got into Cuba and started the first dengue hemorrhagic fever outbreak in the Americas. And that's the one that I've been tracking since 1980s. And we found it so unusual that we were finding the Southeast Asian virus in the Americas, and that's the one that eventually got into Venezuela. It's the same Southeast Asian, and it displaced the American genotype in those countries. And right now, Iquitos, Peru, has American genotype, and in the last 2 years, Southeast Asian has been starting to get in there. They've actually isolated it from mosquitoes, and I think a couple of children this past year. So we're waiting. Poor Iquitos, the population of Iquitos is our experiment. Yeah. Oh, yes.
I have a question about the mice.
Mhm.
Are those mice have a possibility of becoming a stable mouse strain, or do
No.
they have to be made?
Yeah, you have to be made.
So
Yeah,
they're super,
yeah,
super
yeah.
expensive.
Very. You cannot. You can't. I mean, these are. How can you get mice with transplanted, cells?
Like GFP mice.
Yeah, yeah, exactly. No, that's I think impossible to do that. The good thing is that we're amazed that we've actually taken mice sacrificed at day 8 after infection, and their bone marrow is 50-60% human. It's amazing.
That's a feat in itself.
Yeah, it is. But these poor mice, they're very small. They're non-obese, diabetic, skid, gamma radiated, transplanted. Bled. We're amazed that they do have weight loss when they are going through the viremia, but we don't know if it's because of all these things that have been done to them or whether it's the virus. But yeah, my postdoc, he's a veterinarian. He feels really bad working with them because they're constantly dying on them because they're so susceptible to everything. Yeah. Yep. Thank you very much. Thank you.